This is from Mark Majeski, who, as I said, the associate director of another center in Carolina, North Carolina. So this is just one example of many like that. In terms of the stem cell research, I've alluded to Mike Murphy. You're going to hear more about him. He's just recently completed enrollment in 2 trials, back-to-back trials. The second of the 2 trials is oriented towards taking what we learned in the first trial and making it more applicable to office-based practice. So as we work with these stem cells that are your own stem cells, in this case from bone marrow so far, we're trying to figure out how do we take it from a situation that we practice in a hospital like IU, being able to do something within a matter of a shorter time, hour or 2, in a vascular surgeon's office, so that we can ultimately, if it works, spread this, really easily. So this is what the second trial here was about. We'll talk a bit about that. We're currently planning for what's called a phase 2 trial, which is what we call a randomized trial. That means patients will either get their own adult stem cells or no cells, and that will allow us to figure out whether the cells actually work or whether it's just a series of happy incidences, which we don't think is the case, but that's what we always have to test. And then we're planning right now a safety trial for fat-derived stem cells. This is Mike, and you'll see him later. These are some of the cells that come out of fat. Adipose stromal cells, we call them, adipose being a long word for fat. And when we spin those tissues from fat in a test tube, the fat cells go up because they float. It's just like chicken soup. Think the fat comes to the top. And the other cells of interest are at the bottom in the pellet. And that's what we work with, those cells. I've shown you this before. What we found several years ago and published was that provision of those kinds of cells in a mouse with a gangrenous foot, which is designed to be very similar to the disease in people of gangrene of the feet, bad circulation. You provide those cells, you don't fix the whole foot, but it's certainly a lot pinker and much more of the foot is healthy than in the instance where you don't provide the cells and the whole foot really is black and it's soon to fall off. In fact, this finding has been replicated now in at least 7 different places that I'm aware of, and 6 of those have been published. So we have a very consistent finding that this is an important situation. I'm going to skip through a lot of research and just show you something that's a really cool picture. This is the stem cell. We've identified that by some of its surface markers that allow us to color it red. Under a microscope in fat, you see a blood vessel colored green, and the stem cell usually rides on the blood vessel. And that is, we think, actually a general principle, that stem cells are distributed on blood vessels throughout the body. And that was something that we here first described in October, and now a number of other groups are describing that as well. So it's a growing recognition throughout the world. I bring up Dr. Chen here. Some of you have met him. He's our cardiology chief. He's an internationally renowned leader in basic and clinical research in rhythm problems in the heart. He has a special focus and interest in nerve sprouting in the heart, and we have begun to collaborate and have really got a great collaboration going on. Vascular stem cells and how they affect the heart and nerves. Because of that collaboration and our partnership, it was recognized several months ago that the Vascular Center and Cardiology could come together and address a request that the whole campus was making. The chancellor of our campus made a request for proposals for something called signature centers. Called them signature centers because he thought that they would become the signatures, if you will, of this campus over time. And what we were able to do by writing a proposal about an idea that we had, which was to work within vascular and cardiology, to work with adult stem cells, sort of an outgrowth of what I've been describing to you, led to an additional infusion of funding from the IUPUI campus that matched funding that came from Dr. Chen in cardiology. And so was named the Vascular and Cardiac Center for Adult Stem Cell Therapy, or VCCAST, as a new signature center. That's rides on these 3 legs, if you will, the vascular, cardiac, and the campus work. So we're really excited that that has gone to another level, recognizing stem cells in a variety of areas. This is a website that isn't up yet, but this is what it will look like, ultimately. And the point that I want to describe about this website, which will probably be up in the next few weeks, is that it brings together some new research groups that are vascular related, because as I mentioned to you, the stem cells that we're all working with are actually part of the blood vessel. But they're in other areas of the body as well. I've expanded this picture here. Moving from work in peripheral vascular disease and heart failure that I talked about, not very much today, but that we're doing with great interest with Dr. Chen, these areas as a sort of foundation, cardiovascular, we're now moving, using these stem cells from the vasculature, from the blood vessel, into treating, or looking at treating emphysema, stroke, hemophilia, and diabetes. So these are all growing out of this situation. And these are new areas of interest that the Vascular Center is getting into by virtue of the fact that these stem cells are throughout the body. It also turns out to mean that there is a very wide range of importance for these cells. I won't talk about this much today. I'll just point out that it turns out that the stem cells secrete substances that actually protect the brain from injury in a simulated stroke. This is a novel finding and an important one, we think. This is normal brain of a rat. This side was subjected to not enough blood flow and not enough oxygen. And over time, you see how this side has gotten small. It's deteriorated compared to the other side. But when treated with what we call conditioned medium, that is substances that the stem cells secrete, that is largely inhibited, even if you treat after the stroke, which is a really important finding, we think. So we're scaling now and working with others, working with a number of investigators. This was done in collaboration with Yanxing Du in neurology to bring this forward to a stroke center circumstance. And maybe the next year or two, working with Mike Murphy and other neurologists, will be able to bring that forward. We've also found that two stem cell types work together much better than one. Remember I mentioned the inside of the blood vessel and the lining on the outside of the blood vessel are two kinds of stem cells. If you put those together consistently, this is a little, what we call a collagen gel. It's under the skin of a mouse, which is why this looks furry. This collagen gel here, this collagen gel is one stem cell in each case, one type. But if you put them together in every instance, you have this red structure. And why it's red is because it's full of blood flow. So what we found is that the two stem cells work together in a remarkable way. We think that's important also in treating potentially diabetes. Why that is will be described to some degree later on. We're at the outside of those studies with Dr. Molina, who I introduced briefly as a concept before. I'll show you a little bit about him in a moment. But the idea is to use both of these stem cell types to provide blood flow to what's called islets. You may have heard of islets. These are the things that can be transplanted in. Oh, there she is. Transplanted in to patients that don't have enough insulin. But it's important that those islets have enough blood flow. So this is a finding that we think would be important in that regard. And so she'll be talking with you. She's just been recruited to the Division of Endocrinology from Virginia. She's a leader in work for finding mechanisms of how insulin secreting cells called the beta cells develop and are maintained. And we hope to work with her then to make them be better vascularized or bring better blood flow to those so that the insulin can get back out into the body. Postdoctoral training grant is supporting 5 fellows. I mentioned that. 2 trainees have received AHA funding and have accepted faculty positions in vascular research. One of them stayed here. One of them went to Northeastern Ohio. Another trainee is pursuing complementary work as a fellow in pediatric cardiology this year at the University of Pennsylvania. So these trainees are going out and actually, in a variety of ways, doing very well. We're very excited about that. Whitney Seals, I mentioned before, is our most recent trainee to join our training grant, sitting here in the front. She joined us from Albany Medical College in New York. She actually moved in this week. So she's going to talk about research that is going to go on ahead, but hasn't happened yet. In her prior work, in her PhD, her doctoral training, she worked on dietary good fat and bad fat and how it relates to obesity and a certain kind of liver disease that is accompanies obesity. She's going to be working in the lab with Dr. Elmendorf in physiology, along with our lab as a collaborator, on how cholesterol can affect blood vessel stem cells. So our ongoing goals continue to grow and maintain our leadership position with the clinical trials, submit a multi-investigator grant in adipose stem cells. Some people call those program project grants. We also have just recently sent out a letter of intention, we call it, to do work with 2 other key people on campus who are focused on endothelial and hematopoietic stem cells, that's the blood-forming cells as well, So we have hopefully a couple of those. Identify new funding is always important. We continue to need that support because both the infrastructure and the pilots of the high-risk projects I talked about are critical. We can't generally find governmental funds for things that are high risk. It just doesn't work that way. Maintain and develop core functions, continue our training environment, and identify patient opportunities whenever we can. The leg disease future, we see with the fat-derived cells as well as the bone marrow cells that a patient with a threat of amputation comes to the lab. Standard surgical approaches are said not to work. In other words, can't be fixed. Leg has to come off. This is the kind of patient we're looking at. We would envision that a liposuction be done under local anesthesia to get the fat out, prep the cells, and reinfuse them into the leg. And that is what we think Dr. Murphy and others will be doing in the next short time. This is actually the machine, or a machine, that we think will enable that substantially. I like to call it the cappuccino machine for stem cells. Fat goes in the top, literally, and stem cells come out the side. And that takes a procedure that takes us currently about 3 hours and a lot of very skilled technical work to do, down to about an hour and put it in and out. That is one of the elements of sort of creating an office-based procedure, and this is in partnership with a company called Tissue Genesis Incorporated. That's all I have to say. I hope I've given you a picture for some of the areas that we think are exciting